Multiple sclerosis (MS) has long been a mysterious and debilitating disease, affecting approximately 3 million people worldwide. This neurological condition causes a range of symptoms, from vision loss and fatigue to impaired mobility, significantly impacting the quality of life for those affected. For years, the underlying cause of MS remained elusive, leaving doctors to tackle symptoms as they arose in a frustrating whack-a-mole approach. However, recent breakthroughs have shed light on the root cause of MS, opening new avenues for potential treatments and even a cure. The key to understanding MS lies in its mechanism of action. In MS, the immune system mistakenly attacks a crucial component of nerve cells called myelin. This protective sheath surrounds neurons in the central nervous system, facilitating rapid signal transmission and safeguarding the nerves. When myelin is damaged or destroyed, neuronal function is disrupted, leading to the characteristic symptoms of MS. The progression of the disease varies among individuals, with some experiencing relapsing remitting MS characterized by intermittent attacks, followed by periods of relative normalcy, while others face progressive forms where symptoms steadily worsen over time. The breakthrough in MS research came with the identification of the Epstein-Barr virus, EBV, as the underlying cause of the disease. EBV, known for causing infectious mononucleosis or mono, is an extremely common virus, infecting about 95% of the global population at some point in their lives. This prevalence initially posed a puzzle for researchers. If EBV is so widespread, why does only a small fraction of the population, 0.04%, develop MS? To establish the connection between EBV and MS, researchers conducted an extensive study involving blood samples from 10 million U.S. service members. This large-scale investigation was necessary due to the rarity of MS, with fewer than 1,000 individuals in the sample actually developing the condition. The study revealed a clear sequence of events. First, an individual's blood would test positive for EBV. Then, markers suggesting nerve damage would appear without overt MS symptoms. Finally, MS symptoms would manifest. Importantly, no other variable could account for the emergence of MS, solidifying the link between EBV and the disease. Understanding the causative role of EBV in MS development has spurred further research into the precise mechanisms by which the virus triggers the autoimmune response characteristic of MS. A 2023 study published in Science Advances provided crucial insights into this process. The researchers discovered that in some MS patients, immune cells can target both an EBV protein called EBNA1 and a myelin protein known as CRYAB. This cross-reactivity occurs because EBNA1 and CRYAB share a short, identical segment in their structure. As a result, immune cells attempting to control the EBV infection inadvertently attack myelin, leading to the neurological damage seen in MS. The process unfolds as follows. EBV infects B cells, a type of antibody-producing immune cell. In response, T cells, another crucial component of the immune system, attempt to suppress this infection. However, due to the molecular similarity between viral and myelin proteins, these T cells end up targeting myelin instead. This newfound understanding suggests that treatments aimed at either the infected B cells or the overactive T cells could potentially alleviate MS symptoms. Current treatment strategies for MS focus on modulating the immune response to slow disease progression and manage symptoms. These approaches include steroids, used to reduce the severity of ongoing MS attacks by dampening the overall immune response. Antibody-based treatments. These therapies directly target specific immune cells to slow down their response. Medications. Various drugs aim to reduce the frequency of MS attacks and manage symptoms. While these treatments offer relief and improved quality of life for many MS patients, the ultimate goal is to prevent the disease altogether. Given that EBV is the root cause, developing an effective vaccine against the virus seems like the logical next step. However, creating an EBV vaccine has proven to be an exceptionally challenging task. EBV is a complex virus with multiple phases in its life cycle, the ability to infect various cell types, and a multi-layered structure. These factors make it difficult to identify the most effective viral components to target for vaccination. Many vaccine efforts have focused on a surface molecule called GP350, but none have gained regulatory approval. One promising candidate was abandoned when it was found to prevent mono symptoms but not EBV infection itself. However, some researchers argue that this approach might still be worth pursuing, as reducing the immune system's response to EBV could potentially mitigate the risk of developing MS. Other vaccine candidates in development include formulations that combine GP350 with other viral proteins. Additionally, an mRNA vaccine incorporating multiple EBV sequences is currently undergoing clinical trials. These diverse approaches reflect the complexity of the challenge and the determination of researchers to find an effective solution. Even if a successful EBV vaccine were developed, implementing a global vaccination program to truly eradicate MS would be a monumental task. The logistical, economic, and social challenges of vaccinating the entire world population are immense and would require unprecedented coordination and resources. Despite these obstacles, the identification of EBV as the cause of MS has opened up numerous alternative treatment avenues. Some potential approaches include targeting confused T cells. Developing therapies that specifically address the T cells mistakenly attacking myelin could help prevent or reduce MS symptoms. B-cell-directed treatments. Medications like ocrelizumab, which target the B cells where EBV tends to hide, have already shown promise in managing MS. Antiviral drugs. Exploring the use of antiviral medications to suppress EBV activity could potentially slow or prevent MS progression. The journey towards finding a cure for MS is ongoing, and the prognosis is cautiously optimistic. While completely eradicating EBV may be an unrealistic goal in the near future, the wealth of knowledge gained about the virus-MS connection provides numerous avenues for intervention and treatment. Researchers continue to explore innovative approaches, from targeted immunotherapies to novel antiviral strategies, in their quest to improve the lives of those affected by MS. In conclusion, the discovery of EBV as the underlying cause of MS represents a significant milestone in our understanding of this complex disease. While challenges remain in developing a definitive cure, this knowledge has empowered researchers and clinicians with new tools and strategies to combat MS. As research progresses, we can anticipate more effective treatments and potentially preventive measures that could significantly reduce the impact of MS on global health. The road ahead may be long, but the scientific community's dedication to unraveling the mysteries of MS offers hope for a future where this debilitating disease can be effectively managed or even prevented.